Hey guys, so today I'm going to be trying a study trick. So I revised a study trick for anatomy. So I revised my notes from class and I'm going to be reciting them in a way that is hopefully understandable and hopefully helps me do good on my quiz today because I have an anatomy quiz. So hopefully that helps me study and hopefully people watching this video will probably learn a bit or two about stomach digestion and some of the processes on a micro scale that goes on. So here's a bit of my notes. It's a bit all over the place. Yeah. But let's go through them anyways. About 2 days worth of notes. So the first part is about the regulation of stomach activity. So first there's a mix of two parts. There's a churning and mixing, slow movements of food downward, peristalsis story of the stomach of moving ingested food down the digestive tract. Secretions from cells such as mucus, acids, enzymes, and motility. So going into the regulation story, my professor asked the class, what determines the scheduling of what to do and when? Who determines when these things happen in the cells? Or like when are these cells activated specifically? They are first activated in the cephalic phase of gastric regulation. The story of starting, stopping, and responding to current situations. First, the head. Cephalic means head. Head first is the first step of regulating stomach activity because it comes from your brain. It's not your subconscious brain, but it is your conscious brain. Things you're aware of, such as because it's your conscious brain. When you see, smell, taste, or think of food, it is going into the cerebral level or the conscious level. So not subconscious, conscious thinking. Example would be hearing someone crunching on chips. Conscious stimuli of food via the central nervous system or the conscious part of the brain. I'm gonna grab a timekeeper right here. Okay, so the conscious part of the brain becomes the control center or the part of the brain that senses the stimuli. So there's a negative feedback going on. So the receptors we discussed so far were baroreceptors, which detect pressure, chemoreceptors, which detect the concentrations of pH and carbon dioxide levels. So the negative feedback loop, first there is a receptor or the stimulus. So examples would be sight or smell of food or more on that conscious level of sensing things. Then onto a controller or control center of the negative feedback loop. So the pancreas example, blood levels are, if there was, for example, the pancreas, so blood levels varying in the levels of glucose, and the controller would be the cerebral part of the brain sending a response. The effector would be the part of the body that will do something in response to the stimulus, carrying out a response. For the stomach, smooth muscles and glands. There's a change of condition. The negative feedback loop will repeat again from the effector and then back to the receptor. So back on the pancreas example, during this repeating portion, blood glucose levels will start to fall. The stomach has 3 phases. So far I've discussed the first phase, cephalic phase, but the other 2 are gastric phase and intestinal phase. So the brain, stomach, and duodenum all have a say on how completely the stomach activity continues. Stomach activity. Stomach activity is regulated by the autonomic nervous system because it has smooth muscles and glands that are the effectors. Not skeletal muscles, that would be the somatic nervous system. 2 divisions are the sympathetic and parasympathetic. Parasympathetic is the division that turns on digestive process, carries information from the controller to the digestive organs. So from the brain to your digestive organs. We're not talking about we're not talking about blood vessels. So there is no sympathetic division happening here, which sympathetic controls blood blood flow. It tells the smooth muscles to relax and blood vessels to relax. A decrease of sympathetic activity in blood vessels causes the blood vessels to send more blood to the digestive tract. Okay, so sympathetic only takes care of mainly blood to the digestive organs, innervates smooth muscles surrounding blood vessels. Parasympathetic, if it innervates the chief, parietal, and smooth muscle cells, it is parasympathetic. Makes the stomach churn and move for peristalsis. Message carrying of the parasympathetic stimuli from the cerebral cortex to the medulla to the vagus nerve number 10. Vagus nerve number 10 is the major nerve carrying parasympathetic signal to the heart, bronchioles, and digestion. The end of the cephalic phase happens. When the end of cephalic phase happens, smell, sound, taste of food. There's strong emotional events that can happen, located in the hypothalamus of the subconscious, such as rage or fear. Your subconscious. So more onto the gastric phase soon. This is a protective device. It's where fight or flight activates sympathetic nervous system. So the effectors in this story would be, in terms of parasympathetic stimulation, would be mucus. Your head gets your stomach going, so mucus, chief, parietal, and G cells. All start off with parasympathetic innervation before G cells secrete gastrin. So they're parasympathetic stimulation first, then G cells secrete gastrin to activate them. So that chief cells, G cells, parietal cells continue what they are doing. Smooth muscles around stomach start activating. The gastric phase of gastric regulation. So this is the second phase. It has a lot of arrows circling back based on neurally local cues called reflexes. So local cues are called reflexes. Chemically local cues. Local response based in part on neural cues. Extension. Stretch receptors will pick up on the fact that food is reaching the stomach, and the stomach will start a disintegration process. And that's because when there's a distension or enlargement of the stomach, then the stretch receptors will be stimulated for local response. So there's also a local reflex circuit sending stimulus through local nerves to the plexi. Continuing effects. Smooth muscles, parietal, chief cell effects live within the layers of stomach walls. So food as a high pH. We have chemoreceptors which receive information about pH swallowing food with pH of 6 or 7. Saliva and food have an alkaline pH. It is higher than the pH of the stomach detected by chemoreceptors. In a local neural reflex, that information feeds back and causes these things to keep going. Gastrin is a hormone working not through the nervous system, but through the bloodstream because it is a hormone. So that means it's an endocrine. G cells are stimulated by partially digested peptides. Proteins enter the stomach. It is worked on, but some still remain, which are undigested as peptides. If the stomach has not finished its job breaking down proteins, gastrin will keep stimulating cells until their job is done. As long as protein is not broken down, gastrin will keep will stimulate hydrochloric acid and pepsinogen. Okay, so protein-filled food. So in both gastric phase and cephalic phase, there's two possible outcomes. So proteins are digested enough, and proteins can be not digested enough. The third possible outcome would be it would just sit there, so you would have anxiety, lots of worrying, and anxiety, stomach pair situations situations to be handled. Worrying, tight stomach makes the stomach crazy. There could be long-term unresolvable anxiety. Okay, so if the proteins in your stomach are not broken down, all of these things could happen. So there's a combination of events that's happening here. So the cerebrum, cephalic phase, speaks to the hypothalamus, which contains your memories, your worrying, your emotions, and stuff. So the cerebrum has your conscious thoughts, your gastric phase has your subconscious thoughts that affect it. So your hypothalamus speaks to your medulla, and the medulla stimulates parasympathetic activation for stomach processes. The stomach starts to gurgle, pepsinogen acid is secreted, doing all digestive things, but there's no food to work on. Long-term stomach problems, lots of current lifestyles don't mesh with the things that we do, causing gastritis and things like peptic ulcer. Okay, so this example would be the cephalic phase. So there's mental stimulation that can affect it. Sight, smell, food, causing a set of responses. Strong emotional events, things we're born with, and it's all located in the hypothalamus, our memories. Stimuli, so the stimuli we discussed are our thoughts, the smell, and taste. Strong emotions are involved in the fight or flight process. The hypothalamus is considered not conscious or unconscious, but subconscious. So it activates the sympathetic nervous system. Stomach problems, initiating the cephalic phase of the digestive regulation without the resolution of food. The problems would be gastritis and peptic ulcers that would lead to. So stimulation by G cells, if there's a way undigested proteins are present, so if there's protein still in your stomach, G cells will continue secreting gastrin into the bloodstream. Again, endocrine hormone, gastrin. It stops once G cells stops being secreted. So chief, parietal, and smooth muscle cells will all be being secreted by gastrin. So the pyloric sphincter, going down from the stomach, there's a pyloric sphincter. The pyloric sphincter opens to the duodenum. Because the pyloric sphincter will become programmed to allow small amounts of chyme into the duodenum. Chyme in the duodenum is also regulated by the chyme and the food that is left in the stomach. If the stomach has lots of protein, the stomach closes and won't let in food. If the amount of protein is low, small amounts of chyme will be allowed to secrete into the small intestine. The amount of food varies based on how much lipids and carbohydrates are in chyme. Little sampling bits of chyme are being sampled by chief and parietal cells. Decisions are being made by the cells lining the duodenum. So there's 3 in the brain: stomach, duodenum, and the duodenums decide how complex gastric activity continues. So when G cells are not being stimulated, there's no more undigested proteins in the stomach. G cells affect smooth muscle walls as well as the pyloric sphincter. G cells' existence in the pyloric sphincter of the stomach. These cells get involved in the duodenum. The hormone is somatostatin, which has the ability to shut off G cell activity when the protein situation seems to be right. Little sample bits of chyme are sampled by G cells, V cells, chief cells, intestinal wall cells. If lots of protein are left in G cells, then G cells secrete more gastric. The third phase is the intestinal phase, so it's stimulated by food entering the duodenum. Then, to understand the inside food, chief, parietal, and G cells sample the chyme to decide if the stomach should keep going or not. The composition of the chyme determines how fast it goes through the sphincter. So first, the stimulus is received by stretch receptors. Once in the intestine, other cells are involved. Intestinal cells have a alkaline mucus, so the pH is 9. Stomach secretions. HCl is very acidic. Then hydrogen and chloride secrete from the stomach. So we start off with water and carbon dioxide in the stomach, and then it becomes carbonic acid. Then it becomes hydrogen and splits into hydrogen and chloride, which are both secreted from the stomach. Then there's some sodium chloride in there as well. So hydrogen and chloride will bond together to form hydrochloric acid, which is very acidic. So HCl is a stomach secretion. As we know, parietal cells secrete them. And then some of the chloride from sodium chloride binds with some of the bicarbonates, which are formed to form sodium bicarbonate. Sodium bicarbonate forms a basic mucus in blood. Sodium bicarbonate creates an alkaline mucus for alkaline tide. Sodium bicarbonate is used to make alkaline mucus, mainly used to coat the chyme. Going from an acidic solution to a basic solution, it's a switch between the stomach and the intestines. Quick chemical switch between the stomach and intestines. If there's no switch, then you have duodenal ulcers when too much acid is there in the duodenum. Based on how fast chyme goes through the sphincter, what cells are going to be secreting what? So once in the intestine, there are mucus cells, which are very involved with creating an alkaline mucus. Bacteria are able to survive a pH of 2, not a pH of 9. So mucus, very alkaline. Why is the intestinal tract producing a pH of 9 from leftover sodium and bicarbonate ions? Okay. Secrete from mucus cells. Also in the intestinal tract, there's alkaline mucus secreted from mucus cells such as EE cells and enteroendocrine cells, which secrete GIP, gastric inhibitory peptide, which inhibits G cell secretion of gastrin, slowing down stomach activity. Then we have CCK, cholecystokinin, and secretin. Enteric means we're talking about the intestinal tract. So these three secretions will make sure that mucus is secreted with a plane of buffer in it and have alkaline in it. The gallbladder has started secreting its bile. That the pancreas is going to secrete all of its goodies into the duodenum as needed. If the stomach pyloric sphincter is asked to send a little at a time to allow bile to work on lipids before pancreatic enzymes break them down. If more chyme, the pyloric sphincter opens wide. Okay, the response to intestinal phases, G cell and D cell, determines if the chyme is full of fat and protein. If there's a high amount of protein, then gastrin is secreted. B phases tell the stomach what to do when the intestine responds. If there's a decrease in protein, the stomach will start secreting gastric inhibitory peptide. Okay, so there's a constant negative feedback with local chemical signals going on here. So the duodenum is the secretion center of the digestive tract. If there's a high amount of carbohydrates, then you'll get that will get the pancreatic duct going. Negative feedback telling the stomach when to start and when to stop. So E cells stands for enteroendocrine cells. Entero means intestine, endocrine is relating to or denoting glands that are secreting hormones and other products directly into the blood. Exocrine goes into the lumen of the tract, not in blood. Gastric regulation, as a recap, we went through all of the phases and intestinal phase. The intestinal phase is what is the stimulus. So think feedback triangle and regulation. So the stimulus for the intestinal phase to kick in for the duodenum to have. So this is the third phase, the intestinal phase. The third phase is the stimulus for the intestinal phase to kick in for the duodenum to have some effect on what the stomach is going to do next. So how does the duodenum know it's time to tune in and speak to the stomach? It has the stretch receptors in the duodenum during the receiving of chyme through the pyloric sphincter. The stretch receptors become the main stimulus for the intestinal phase, the distension or enlargement of the duodenal wall. The controller is the local reflex from the brain and response being sent from the brain, the neural plexi, the myenteric and enterogastric message being sent from the brain and response being sent from the brain. So, stretch receptors are playing an effect for sensing the stimulus, which is distension or enlargement. Effectors, so there's multiple effectors. Once the duodenum knows where there is food coming into it, G cells and D cells will decide / trigger whether to stop or continue stomach activity. G cells will secrete gastrin to continue stomach activity. D cells will secrete somatostatin into the lumen of the duodenum. Why? It's because it is secreted in the bloodstream. They are hormones, gastrin. They go into the bloodstream and are picked up locally by G cells, Purah cells, and smooth muscle cells. And all of them continue the stomach activity, or D cells secrete somatostatin as an endocrine hormone going into the bloodstream. It is picked up by the same cells of the stomach and inhibits stomach activity. Mucus cells are also effectors. E cells, enteroendocrine cells, all of these stomach cells, effectors, are all receiving a signal from the neural complex of food entering. So there's a situation of if this happens with chyme, then this happens. Chyme. If chyme is full of polypeptide strands of amino acids very strongly together along primary structure, what is going to happen? If chyme still has lots of polypeptides, who is the effector and how will it respond? G cells will respond by producing more gastrin. So the effectors are mucus cells, D cells, G cells, pyloric sphincter, muscle cell effectors, duodenal mucus cell, and effectors secrete mucus into the duodenum for chyme to come through, and also the EE cells. If the chyme is not full of polypeptides, but high in lipids and carbohydrates, who is going to respond and be the major effector? EE cells are going to start secreting GIP, gastric inhibitory peptide, CCK, which stimulates the gallbladder for when lipids aren't broken down and you have a fatty diet. CCK, so carbohydrates and lipids breakdown. Okay, so what does the pyloric sphincter do with a fatty diet coming through? Sends small amounts through at a time because it takes a while for bile to do its thing before the enzymes can get to them, slowing the flow into the intestinal tract so that the gallbladder will have chyme. Having a low pH in the stomach indicates that, what does it indicate about stomach activity? This means stomach activity is finished, it has done its job, and acidified the whole thing. E cells secrete gastric inhibitory peptide, thus secretin. Why secretin, you ask? In addition to slowing down stomach activity, Secretin will do what for the duodenum? Secretin stimulates the pancreas to secrete neutralizing or alkaline or buffering pancreatic juices. Also inhibits gastric activity, stimulating bile. EE cell hormones from the cell in the duodenum. Oh. So EE cell hormones include secretin, which speaks to the pancreas, which secretes buffering alkaline juice. CCK stimulates the whole buffering activity as well as GIP. VIP, vasoactive intestinal peptides, activates blood vessels in the area to dilate and get ready for absorption. GIP, gastric inhibitory peptide. So gastric regulation has a control system, nervous system is the initiator, brain locally through the intestinal plexi. Responding to nervous signals, endocrine hormones are secreted, gastrin, somatostatin, CCK, etc. GIP, secretin also, that stimulates exocrine hormone secretion. Exocrine hormones go into the lumen of the tract. HCl, these include, for exocrine hormones, HCl, pepsinogen, mucus, alkaline buffers. Does not go into blood, it goes into the lumen. Pancreatic juice and also pancreatic juice. These are not hormones, they are digestive juices. So there's a pattern going on here, nervous system, it triggers endocrine system. So the intestinal tract is the site of most chemical digestion, secretion, and absorption. It has 3 sections of proteins. The intestinal tract is divided into the duodenum, jejunum, and ileum. Approximately 12 finger widths. Microscopically, you can see a difference in the size of crypts and villi. Crypts versus villi, so we have deep crypts in the duodenum for intestinal gland secretions, and short villi, so not much absorption. The intestinal tract has villi are used for absorption, increasing surface area. The intestinal tract has cross bands of tissue, circular folds called rugae. Bones, but here in the duodenum we have plicae, which are intestinal tract that runs transverse. Okay, that's enough for today. Let's take that quiz. Hopefully this helps you guys understand some of the hormones, some of the parts of the digestive process that goes on in the stomach, duodenum, and digestion.